Good morning. It is wonderful to be here. I came today just for this meeting, which is means my personal body clock is a little bit off, and I will certainly try not to pretend that it is not 6:30 or 5:30 in the morning. Helen asked me to speak on this very narrow topic of where will cavernoma treatment be in the next 20 years. So, and then I said, honey, my talk is about 50 minutes long, and she's like, no, 45. You got to leave time for questions. So I will probably talk American fast and stop me if you need me to. But the first thing I want to share with you is the reason that I started the Alliance to Cure Cavernous Malformation in the first place. This is 20 years ago. There was a little girl. Her name is Julia. I happen to have given birth to her. Before this, or at the time of this picture, she had already had 4 brain surgeries for hemorrhaging cavernous malformations, or cavernomas. She was born with a genetic mutation. We didn't know what it was at that time. The gene hadn't yet been identified, but it since has been. It's a CCM3 mutation, which caused her to have cavernomas, I'm going to get it right by the time I leave today, cavernomas all over her brain. Her first surgery, she was 4 months old. And even at that point, she already had 17 lesions. I was a clinical psychologist, and I wasn't, because of the series of medical events, able to practice anymore. It wasn't fair to my patients for me to be stepping in and out in that way. So I was home, and like Ian, I had nothing better to do. And so in 2002, I started the Alliance, what was then the Angioma Alliance, and has since in 2022 become the Alliance to Cure Cavernous Malformation. And the reason we switched the name of the disease is because of the official name in the ICD, International Classification of Diseases, version 11, we're included, and it is called cavernous malformation. So my why is now this person. She is 26 years old. She is disabled, but you can't tell. She has invisible disability. This is her at a fundraiser just a couple weeks ago for the Irish Music School, where she sings and dances in Virginia. They made it look very, I think, pretty authentic. And she has other passions, horses and dogs, and et cetera, et cetera. So she's living a good life still with me, and looking forward to coming back to Ireland just this next summer. So in 2005, there was a young gentleman from England who reached out to me by email. And I wasn't all that good at answering emails back then. I'm still not great, but I've gotten a little bit better. And I think I might have answered his first email with something very short. His email to me was, hello, I would like to start an organization in the UK, and can you give me advice? And I probably said, just go for it. And so he wrote me again, and then he wrote me again. And you know, I got the idea that this gentleman was probably pretty persistent. And lo and behold, Dr. Stuart started Cavernoma Alliance UK, which at that point was called Angioma Alliance UK, as a sister organization to what we had going on in the United States. And this was from the newsletter that our organization put out announcing the formation of Angioma Alliance UK. Several years later, the UK Board of CA UK Angioma Alliance UK Board of Trustees reevaluated the name because nobody here knows what an angioma is, and at this point nobody in the US knows what an angioma is either. The only reason it was the name in the first place is alliteration. And so Cavernoma Alliance is in deference to the Alliance to Cure Angioma Alliance, and Cavernoma, of course, is the name. What he did was amazing, and Helen and Heather have already shared so much of what Ian accomplished during his tenure as the founder, and he never wanted to call himself the CEO. He was always the coordinator of CA UK. And what I would like everyone to do is just offer yet another round of applause, because he was having you applaud for everybody else. Please let us applaud for Dr. Ian Stuart. Who is also a friend and whom I love dearly. So now, the reason that I'm here, what is a cavernoma? It's a blood vessel abnormality, and all of you in this room know this. I just want to level set here to make sure we all are speaking with the same idea of what these are. They're abnormal blood vessels in the brain or spinal cord. They look sort of like mulberries, and they have not tight junctions between the cells of the blood vessels. And so they are leaky. They intrinsically ooze blood. They can cause seizure. They can cause stroke, hemorrhage stroke. They cause headache. We are told all the time, oh, your headache has nothing to do with your cavernoma, but we have learned better in the meantime that people with cavernoma tend to have headache more often. And they cause functional neurological deficits that sometimes are not directly related to a hemorrhage. They can come either in a hereditary form, known as a familial form, caused by any one of three genes mutating, CCM1, CCM2, or CCM3, the creativity you can sell right there. Or they can be sporadic, meaning people have just one, the mutation is in the lesion itself, not throughout the body. And once that lesion is removed, frequently that is curative, but for many people that is not an option. So I'm going to look back 20 years before we look forward 20 years in order to give some context. So in the basic science realm, I want you to look at these carefully and memorize them because there will be a quiz at the end. In the basic science world, though, this is what was happening 20 years ago. The picture on the left is what we understood about cavernoma at that point. We, in 2006, had identified all of the three genes that can cause the disorder. And we had a basic idea of what the neighborhood was that these genes live in. And the little ovals that you see are other proteins. And then you can also see there's a whole bunch of question marks there. The scientists put those there, because that is exactly what they were thinking, huh? And we don't know what happens there. So what you want to focus on is the busyness of these pictures. The busier they are, the more that means we understand not just who the players are in this field, but also what each of them does, what position they have. On the right, you see a little bit further along in 2012, the picture is busier. We know more of the players. We know what roles they play. Then finally, this is where we are today. We've discovered color. And we also have a very busy picture that includes many, many different players, many proteins. Each of those ovals is a protein. The connections are the pathways that they work in. And the interesting part here is that any single one of those could be the target for a treatment that could make a difference in the disease. And so we have a lot of drugs out there that are already targeting these proteins, these pathways. We now have a list of medicines that is that long, and I'll go through some of them as potential treatments for the disease. So the other thing that we needed to develop in the last 20 years were ways to model the disease that were not human. We're not going to test every drug in human beings because that just isn't efficient and it can be harmful. So we needed to understand, in order to get those very fancy pictures, we needed to understand what was happening in models that were not human. And one of the most basic ways to do this is in endothelial cell lines. And the endothelial cells are just the cells lining the blood vessel. And, and I see David White and David, forgive me for anything I say that is not accurate or true. I'm looking to him and feeling intimidated. So endothelial cell lines are just cells, that single layer of cells. They give you just a very, very basic idea of what's happening. Our next more complex model is C. elegans, which is nematodes, worms. We love our worms. We love our worms because They're very fast reproducers. They're very simple creatures. They have just 900 genes, 900 odd genes. The cavernous malformations, once they developed the models, have actual changes in the single tube, one of the two tubes that runs from end to end of these worms. And you can actually see, I don't know if you can see them here, but there's like a little bubble in the tube that indicates where there's a problem, and we can fix them. So there are times where we've been able to apply treatments to these nematodes and watch how the worms recover. David will tell you more about Drosophila than you will ever want to know. Then you will find it fascinating, all about the flight path of the flight mechanism for the Drosophila. We use them for CCM research again because they reproduce quickly and it's easy to get some answers from them. And then our more complex animal, and we consider zebrafish more complex because they're a vertebrate. So it's the first animal in the line that has a vertebra, is used because the embryos are see-through. And as they develop, you can watch it in real time. That's unlike any other creature that we typically would use in research. When we first developed the zebrafish models, they showed the disease by having an enlarged heart. There was no cavernous malformation lesion in the beginning, back when they were working with these around 2008, 2010, even 2012. It wasn't until really the last 10 years that they were able to develop models that actually showed cavernous malformation lesions. Then we have our mice. The mice are the bread and butter. This is where once a treatment shows itself to be effective in a mouse, that's when we know we can start taking it into people. And this mouse model has developed over the years as well. When we first started, our mice had cavernoma all over their body. When they knocked out the gene, they knocked it out so that it was in the organs, everywhere. And these mice did not live very long, and not because anything was happening with their brains. It was that their bodies just couldn't manage that burden of lesions. So then we developed a better mouse, and the better mouse had only brain lesions, but they were only in the back of the brain, which is good. The mouse lived longer, but it wasn't exactly what the human condition was like. So finally, in the last, I would say 7 or 8 years, we have a mouse that has lesions throughout the brain. They can live relatively long. You can do a lot of science with them. And when you see improvements in these mice, you can be pretty confident that this is something that we should be at least looking more seriously at for humans. Okay, so that's where we are right now. We are moving forward, and there are now 3 labs, 2 in Europe, 1 in the United States, who are creating these models that are organoid models. And what is an organoid? An organoid is human tissue. It is not just that single layer of cells, of endothelial cells that we see in cell lines. It's actually those cells plus a lot of the supporting layers of cells, the astrocytes and parasites and all of these other sites that they talk about in the cell world. And so they give a much more accurate idea of whether or not a treatment is going to work than you would see almost with any of these other models, and you can screen quickly. So they have the benefit of fast and the additional benefit of close representation. And there are other tissue models as well that are in development. Again, the Swedes just recently got a large grant to develop this. In Greifswald in Germany, we have commissioned them to do some drug screening for us. And then there's a group in Cincinnati in the United States that are doing this. And then finally, the thing that all of us are talking about now, right now, is AI models. And AI will be useful as it becomes more developed for helping us understand what potential treatments are. Certainly, it's already being used to mine the literature to see if we're missing something. In the very first AI models, for example, it pulled up that strychnine could be a really good treatment for CCM, except, maybe it's not a good idea for humans to ingest it. So we put that one aside. AI is not perfect, but it could, in the end, give us some things. The other thing that we learned over the last 20 years is what kind of lifestyle changes or adjustments we can make to perhaps modify the disease course in preparation or before we have some real types of treatments. So what we thought in 2005, and what I listed on my website, is that these are the things you need to be worried about. Blood thinners. Don't take blood thinners ever. Don't take them because if you thin the blood, then your hemorrhages are going to you're going to start hemorrhaging and your hemorrhages are going to be bad. Worse than if you weren't on blood thinners. We thought that pregnant women had more hemorrhages than people who were not pregnant, and therefore pregnancy was a risk. We thought exercise might be a problem if you get, Valsalva movements, or if you just get your heart rate up too high. Skydiving, scuba, inflammation, especially the kind that come from smoking or from flu that's systemic. So that's what we thought in 2005. Now, we think, you know what, blood thinners are probably okay. When we look back at, years and years and years of literature, people who have been on blood thinners for other reasons don't hemorrhage anymore, and in some cases, they hemorrhage less. And so someone here eventually will be talking with you about an aspirin study that is being considered. And that sounds like, maybe not what we would have expected. But if you think that maybe the cause of hemorrhage is blood clot, then it starts to make sense. So we're learning over time. This is the process of science. Science is never wrong. Science just learns more over time. Pregnancy now, it doesn't appear that there's any greater risk to women being pregnant than when they're in the not pregnant state. Aerobic exercise seems to not be a problem. The jury's still out on strength training. We just don't have enough data. Where we do have cautions now are oral hormone use, and it's just oral, so hormone replacement therapy, oral contraception. Women who take these agents have a higher risk of hemorrhage. So there are other choices, and we can, you can certainly talk with your doctor about them. I am not the person to talk with about them, but the research at this point is showing that this is an area where care is needed. And again, just oral forms. Low vitamin D is an issue. We need folks to supplement vitamin D, especially here in England. If you're not getting enough from the sunshine, then you should be taking a supplement daily in order to make sure that your vitamin D level is at a normal level. Preservatives and emulsifiers in food. This was 2017 research, so relatively recent. We discovered that preservatives and emulsifiers have an impact on the gut lining. We always knew that, but that impacting the lining of the gut causes an inflammatory response that can impact the lesions in the brain and make them proliferate, so you get more or hemorrhage. That we didn't know. So it's very, very important if you have a CCM3 mutation that you change your diet, because they have intrinsically an impairment in the gut lining. But even if you don't, you can make that same impairment happen by eating a whole lot of very overly processed foods that have a ton of preservatives or emulsifiers in them. And we have a lot of information on our website about that. And I do think most of what we have applies here in the UK as well. And then finally, inflammation remains a concern now. We didn't have COVID in 2005. We continue to tell people that getting vaccinated is a good thing, that you are more at risk with COVID, having that disease, having that illness, than you are at risk by having the vaccine. And you are at risk in a longer term way. So flu and smoking again. Where were we in 2000? I didn't even bother with a 2005 slide here. Because I did. I started one and had a goose egg, someone holding a goose egg, and I thought, oh, that's just weird. So in 2005, we had nothing. There was no pipeline of medicines, no idea as to what could be helpful. So I'm starting here in 2019. So this is just 6 years ago. Just 6 years ago, we had atorvastatin, a statin drug, that had just begun a clinical trial in Chicago with patients. So as I go through this, preclinical means no patients, no humans have taken it yet. Phase 1 means that healthy volunteers, not people with cavernoma, but just healthy volunteers have taken this and it has been deemed safe, or that's why they're taking it, is to make sure that it's just safe for human consumption. Phase 2 is the first time that a cavernoma patient will take a medicine. Those are usually small trials, and they're trials to make sure it's safe for cavernoma patients and also to figure out if the dosage that is being chosen for a trial is the correct dosage. So we have atorvastatin just starting its first in cavernoma patient trials, and we had propranolol being tested in Italy. Propranolol is blood pressure medicine. Statins are cholesterol medicines. These are two heart-related medicines that were the first things repurposed for cavernous malformation, for cavernoma. And then we had 2 industry partners developing drugs that were specific for cavernoma and not to be used for any other disease, at least in the beginning. One of them had the same mechanism as atorvastatin, BA1049. It was more specific to CCM. It wasn't going to lower anybody's cholesterol. And then the other one is REC-994. A company called Recursion was developing this. It was an antioxidant that was of a type called superoxide dismutase. You don't have to memorize that. There really won't be a quiz. That's where we were in 2019. This is where we are today. And this is how you get from a little at a time to a lot all at once. So we went from having 4 drugs that were under consideration to now having this whole raft of treatments that I will go through. So you can look at them, but I will walk you through them. That's part of my job of being here. So BA1049, that Rho kinase inhibitor similar to atorvastatin that was being developed for CCM specifically, was sold to a company who does clinical trials, Neurelis. They are very close to starting first in CCM patient trials. They have already done their first in human safety trials, so they'll be moving it into patients probably in the next year. They're trying to do a lot of pre-work to make sure that the clinical trial they design is the right design. Propranolol finished its trial. It actually looked like it had some effect. The trial was small. It didn't have a placebo arm, which is problematic. It had very broad inclusion criteria, but it did look as if it had some impact on the number of lesions that formed, and also that it may have had some impact on stabilizing the lesions. The problem with propranolol is that it's generic, and there isn't a company right now that's willing to do any more with it. There is some company in the EU, I think it's the EU, Helen, EU, that is perhaps interested in reformulating the drug to make it more specific to CCM, but right now they're doing more due diligence, looking at the medical records of patients who have cavernoma and were on propranolol to see if they can find something in medical records to justify this. We also have a pilot study happening at a children's hospital in the US. Kids who show up with a brainstem hemorrhage will get propranolol before their brain surgery. So the intervening time between the hemorrhage and the surgery, with the hope is being that they will not have a second hemorrhage in that intervening time. As I mentioned, low-dose aspirin is under consideration for a clinical trial. It is probably nearing phase 2. There are these other drugs now, as I move down, that, and this is where we're talking about not more distant neighbors, but actually closer neighbors to CCM in that one of those big mappy pictures. But these are beginning to work themselves down into drugs that have higher side effect profiles. So we have these 2 PIK3CA inhibitors. PIK3CA is a different protein, but that when it's mutated at the same time as the CCM genes are mutated, the disease, CCM disease, becomes more aggressive. So those lesions grow fast and hemorrhage more. And so the thinking is if we inhibit PIK3CA, that we will still have cavernoma lesions, but that they'll be quiet and they won't do anything, which is not a bad outcome. For alpelisib, it's a drug that's already on the market. It's not off patent yet, so there is a company behind it. At Texas Children's Hospital, a few kids who have had their cavernous malformations, one of them removed, but have the familial form, so they have more than one. Those ones that were removed were analyzed to see if they had a PIK3CA mutation. There was one found. And so those kids have been put on alpelisib to see if they can keep the remainder of their lesions quiet. It's a small trial. It's a pilot. It will be a while before we learn anything. But in the meantime, this other company, Relay, is exploring CCM as an indication for their brand new, not yet on the market PIK3CA inhibitor. And they have suggested to us that they will include us in the second phase of their phase 2. So they have phase 2A, 2B, and 2C, and we will go into phase 2B. And that's a story yet to be told. We have this other group called mTOR inhibitors, which are also in that oncology realm, but are used more broadly for other diseases, have less of a side effect profile than PIK3CA, but certainly more than statins and beta blockers. Rapamycin is one of them. Sirolimus, you may have heard of as an alternate name. There is a small trial underway in China right now for this drug. And a second mTOR inhibitor, whose name I cannot reveal because it's still confidential, a trial is planned right now in China for that and should be starting in the next year. So now we're going to move out of drugs and into the other kinds of treatments, out of small molecule drugs, out of pills. Genetic therapies. CCM3 gene replacement is being researched very seriously here in the UK at UCL and in the United States at Duke University and the University of Chicago working together. Those programs are moving their way along, and everyone is involved in them on both sides of the pond, are very excited about what they're seeing. There is ASO, which is antisense oligonucleotide. Gosh, I thought I was going to get it this time. Antisense oligonucleotide treatment. A new company to us, Cantera in Denmark, is developing this type of treatment, which is genetic. It probably is targeting PIK3CA or mTOR because it tends to target genes that there's too much of, or proteins that there's too much of, and that's what the case is for CCM. Those two are. They haven't told us. They won't tell us. It's all very proprietary, and we'll find out as things develop. CD38 monoclonal antibodies are used right now in multiple myeloma, and the thought behind them is that they trigger an immune response that allows the body to almost heal itself. So this trial is looking to see whether or not the introducing CD38 monoclonal antibodies will trigger the same immune response that will have an effect on cavernoma. Focused ultrasound, I'm going to talk about a lot more in my next slides, but that is focused ultrasound like what you use to see your baby boy or baby girl in utero, but targeted at a single place. And then REC 994. REC 994 was on my last slide. It is that antioxidant, and we have. That's okay. I want to check something here. Okay, so the REC 994 trial ended in spring, and it ended for a variety of reasons. One is, and I believe the major one, is because the stock market pretty much crashed in March and April, and the company developing this was not able to sustain all of its pipeline of drug trials, and half of them were stopped, including ours. The trial at that point the phase 2 trial had been going on for 3 years, and there was some improvement seen in people who were taking the highest dose. There were people on placebo, And then there were people on 200 milligrams and 400 milligrams. And we think that at least for the first year, the people on the 400 milligram dose were seeing a reduction in lesion size and less hemosiderin, so that iron deposit outside of the lesions, meaning that probably the vessels were more stable than they had been. But the trial could not be continued. So I'm going to check my slides because now I don't remember if I'm talking. Nope. So what has happened? The trial has ended, and we've had a relationship with that company for a very long time, when the founder of the company was still a graduate student at the University of Utah. And so my daughter was patient number 1 in the trial of everybody because of the relationship, and because I wanted her to be the beta tester, and if anything went wrong, I wanted to go wrong and have with her so that we could fix it for everybody else. But what they're doing is giving us everything, giving the Alliance to Cure all of the leftover drug, both the drug that's are still in kits ready to be handed out for trials. We have enough to run a whole year worth of clinical trial. All of the powder that hasn't yet turned into pills. There are like 200 kilos of powder. It's crazy. We don't have the money to be able to turn them into pills, but good for us. We've got it. All of the permissions from our Food and Drug Administration to run a trial, and the orphan drug designation, and then all of the data, which is the most valuable part. So our role with this will be to reduce the risk of bringing this back into trials. We can look at the data more carefully. We can try to bring back some of the patients that were in the trial, because we know a lot of them, and try to get more information from them. Like, what is their blood type? We're seeing now that blood type might have an impact on whether or not people have ischemic stroke. This may indeed be something. For people with cavernous malformations, we don't know. So maybe some responded based on blood type versus others. There could be a whole host of things. I just threw that one out there. We're also going to be testing the drug in mice at other doses, and also in combination with other medicines, to see if a combination drug might be even more effective. And if we can get the risk down, we should be able to find a partner back in industry who can take it back into trials. So although the story seems like it's over, it's not over yet. So this is our 20 years of Cavernoma progress, the end of my looking back. 2005, CA UK, then Angioma Alliance UK was founded. All 3 genes were identified by 2010. The first potential treatment was identified in 2009, and that was statins. And as a result of work that Rustam Al-Shahi-Salman did, the word hemorrhage finally had a consistent meaning. He organized a consortium of our science advisors, the UK science advisors, they all got together and said, all right, what does hemorrhage actually mean, and how can we define it, and how can we be consistent across studies so that we can compare apples and apples? That was a big deal, and it still stands today, that definition. 2010 to 2015 is when we really had that peak of preclinical research that made those pictures get all the way to color. And we understood then that CCM3 is a somewhat different mutation in that it works in multiple pathways, some of which are not involved in CCM1 and CCM2. 2015 to 2020, we kicked off our first clinical trials with atorvastatin and propranolol. We had surrogate imaging and blood biomarker work beginning, and I will tell you in a moment what those things are. The microbiome was implicated. That's when we first learned about emulsifiers in the gut. Many potential treatments were identified. And now, 2020 through 2025, we've got 3 trials already finished. One of them had some positive results. The atorvastatin trial had a neutral result, didn't seem to cause any damage, but also didn't seem to help. And REC 994, probably somewhat positive, more to come there. We began to understand the role of PIK3CA in making the disease aggressive, the role of DVAs, developmental venous anomalies, in increasing the risk of developing more than one cavernous malformation, even in the sporadic form. Focused ultrasound was identified as a potential treatment, and gene therapy research begins, and this is where we are now. So this is quite a distance in 20 years, and I'm proud to have been part of it. I'm hoping that you all are proud to have been part of it. So moving forward, this is where we get to talk about that. I have a crystal ball there. It's a crystal ball. It truly is a crystal ball, because when we're talking, that cone of uncertainty gets really wide by the time we get out 20 years. So we have challenges right now that need to be overcome in order to get treatments to people. The challenges are in diagnosis and disease monitoring and in targeted delivery. And we also have some potential cures that we are already doing research on. So I'm going to talk about diagnosis and disease monitoring challenges. As you know, patients are often not diagnosed until they have a hemorrhage, which is like the worst outcome. Why would you want to wait until a hemorrhage before you have the diagnosis if there is a potential for you to have a treatment that could prevent it in the first place? Now, we know that hemorrhage is very, very rare, but if you know you have a lesion, certainly if it were me, and there is a safe and effective treatment, I would probably want to avail myself of that. So this will become more and more of an issue. Hemorrhage itself is too infrequent, as I mentioned, to be a clinical trial endpoint. In order for a clinical trial to say, we have reduced hemorrhage directly, the trial would need to have hundreds to thousands of patients in it, and it would need to be very long. That is just not doable under the financial conditions that all of us live under, and that industry is willing to take on. So we need alternate ways or surrogate endpoints for measuring whether or not a drug is having an effect. So that's things that will lead us to believe that they reduce the risk of hemorrhage. It could be biomarkers in the blood. Right now, the thing that's being looked at most seriously are metabolites in the blood. May change depending on whether or not you are at high risk for hemorrhage. There's also imaging biomarkers. So the picture that you're seeing is a qualitative susceptibility mapping picture. This type of MRI sequence allows you to see the exact amount of iron, hemosiderin, that's been deposited in a lesion. So that gives you an idea as to how active the lesion has been, with the idea being if it gets more active, you are more at risk of having a larger hemorrhage. These are our challenges. None of these have been qualified for actual clinical endpoints by any of the regulatory bodies that decide whether or not a drug can be brought to market. So what do we have here as opportunities? We can begin improving diagnosis, and we're working on this in the US, by tagging, looking retrospectively at people's medical records, folks who have already been diagnosed, to see if maybe there was a hint somewhere earlier in their medical history that could have flagged them to go for MRI to get the diagnosis. Maybe there was a history of headache in a certain pattern. Maybe there was some other, a Bell's palsy or a weakness or something, and something that looked like epilepsy but was never really diagnosed, that we could go and then go into existing people's medical records and discover whether or not they should be moved to the next step for an MRI. We also can continue to develop these blood biomarkers, and here is this novel thing that I just learned about in the last couple of months called a biosensor, similar to continuous glucose monitoring, where perhaps you could get something going around in your blood that could be reading out whether or not the specific metabolites or proteins that they're looking for to indicate that you're at higher risk of hemorrhage are changing, and then you could get an immediate readout. And again, we're talking in the future, not next year, but in the distant future. We could continue working on identifying short-term predictive changes on imaging. We're applying for a grant right now to look for monitoring imaging biomarkers. We look for diagnostic and predictive. Now we're looking for changes in the short term. Better CT scans perhaps assisted with AI to be able to tell whether or not something is a cavernoma versus something else, and then trying to increase the speed of MRI so that our kids can get MRI and have it be a high-quality MRI without being sedated. The next challenge is targeted delivery. This is huge. I mentioned that some of the drugs that we're looking at are oncology drugs. They are toxic. They're toxic. They're designed to kill tumors. They do not have to pass safety tests. They are not intended for chronic consumption. But they could be cures for us. So we weigh this. How do you do this? So what we really need are a way to deliver these drugs directly to the cavernoma lesion rather than have them metabolized in the digestive system, or even directly injected by an IV, because even if you do an IV, the drugs, if you don't have them somehow targeted, drugs wind up in the liver and other organs. So we need to find a way to deliver. What's going to happen is, first, drugs will be delivered to all of the brain blood vessels in a targeted way. That is what they're working on right now and actually making a lot of progress on. Next, we hope that we will be able to target the cavernous malformations, the cavernomas only, so that only there a drug will be released and only that will be treated. We already have some research happening. We're working with University of Pennsylvania on a lipid nanoparticle project, with another group, the Stanford Research Institute, on a peptide-based molecular guidance system. Gene therapy is being delivered via viral vectors. They don't actually have active virus in them anymore. They're just kind of the shell of the virus bringing things into the cell. And then we have focused ultrasound that can help to get things into the cells. The challenge here is that when we are looking at approval, every single pairing of a delivery system and a drug needs to go through its own trial. So I can have the best lipid nanoparticle delivery system for rapamycin in the world, but if I want to swap out alpelisib, I need to start the trial over. Even if I know alpelisib works and it's approved, even if the lipid nanoparticle works and it's approved, once I combine them, it's a whole new thing. And that's where this could become very unwieldy very fast unless we figure out a different regulatory pathway for these types of delivery opportunities. Focused ultrasound, this is like my baby. This is what I am spending a lot of my time on in the US. So as I mentioned, ultrasound waves are just like what you use when you're pregnant and you want to see your baby girl or your baby boy, but they are targeted at a single location, in this case, the cavernoma. The focused ultrasound can be used in two different ways. It can actually raise temperature and do what's called ablation. It can heat and destroy tissue. That's used in cancers frequently. But it can also be low intensity. So the waves move very slowly, and it can be combined with these things called microbubbles, little gaseous bubbles that are put in your blood vessel through IV, and they, with the focused ultrasound waves, bang up and down against the walls of the blood vessel. In the places where they're banging up and down, they're opening the blood-brain barrier just a little bit temporarily. And you're thinking, oh my God, if I open the blood-brain barrier, doesn't that mean I'm going to have a hemorrhage there? And that's not the case. They do not cause hemorrhage, at least not in the mice. And in fact, what they found in the mouse studies that they did at the University of Virginia is that where those microbubbles bounced, a wound healing response started. The immune system activated, and the lesions started stabilizing. In some cases, started getting smaller. In the baby mice, where there was sonication, that's what this is called, those areas didn't develop lesions at all, despite other areas of the brain developing them. And then when the cells regenerated in lesions, they were normal looking. They never turned into cavernous malformations in the adult lesions. So we're looking at this as on its own a possible treatment for cavernoma. And we are hoping to start a trial very soon. I'll tell you that in a minute. But the other thing that it can be useful for is because it does have the ability to open the blood-brain barrier a little bit, is you can connect, you can have those microbubbles be a delivery device all on their own, and where the focused ultrasound hits them, that's where the drug or whatever is delivered. Not something you want to do every day, so if this is a daily treatment, probably not going to have a focused ultrasound machine hooked up in your house, but if it's a one-time thing like a gene therapy, this could be an alternate delivery device. We are working right now to start a phase 1-2 study, and in this case, one is patients. Phase 1 is patients. It's just the safety part with adults at the University of Virginia, which I'm really hoping will start already in late 2026, late next year. Again, like with targeted delivery, lipid nanoparticles, all of those things, every brand of machine, Alex is back there saying, every machine? Because he questioned me on this yesterday. Every manufacturer obviously makes a few different machines. So the Ford Mustang would need to be would need to be approved separately from the Dodge Ram. So we have each manufacturer will have like 3 different machines that might be able to do those. Each of those needs to be independently tested for the disease, and then each manufacturer needs to have its own series of tests. So right now we're working with one company, one machine, but there are other companies out there, and their machines will also need to be put into trials. This may become the treatment of choice for people with sporadic lesions very quickly. If we find out that this works within the next 5 years, this could be what you do. It may also become the treatment of choice for people with aggressive familial lesions. Get rid of that one lesion, calm it down, perhaps even make it shrink and go away. Eventually, these machines will also sonicate larger areas of the brain. There are scientists working on this right now. They are starting with glioblastoma, and then hopefully they can move to us. And then we also need pediatric trials, and we're already talking with pediatric facilities in the US. This machine exists in the UK, and this will be technology coming to you. And even if it doesn't come to you, if you save up your pennies, you can fly to the US. It's a one-day overnight thing. You can come, give us your money, we'll give you the treatment, you can go home, come back in about 6 months, make sure everything's good, and then you're free to go. Just saying. All right, last section for me, gene therapy. The work that's happening currently is gene replacement therapy. And what that means is that a whole new copy of the CCM3 gene is being inserted into a cell, and that copy gets read so that the CCM3 protein, also known as PDCD10, is produced. That is problematic for a couple of reasons, or challenging. CCM3 is the only gene that's small enough for this technology of the 3 genes. The CCM1 gene is enormous. The CCM2 gene falls somewhere in between. The only one that we can use replacement therapy as it exists right now is delivered by viral vector technology, is CCM3. But it's a good place to start. CCM3 is more aggressive. It affects our kids. 50% of kids with CCM3 will have hemorrhage under the age of 18. These kids develop all kinds of other issues too. We do not know yet, at least in the American study, if this therapy is going to help adults. We're pretty sure that it's going to prevent the development of new lesions, at least for a while. That's the other part. We don't know how long the treatment lasts. We don't know yet whether or not the lesions will regress. And that is a story that's going to be unfolding over the next year. So what's an alternative? An alternative for everybody else is gene editing. You can make precise changes to a gene. CRISPR is the technology that's used for. All of you have heard of that at some point or another. But the problem there is that each of those CRISPR experiments needs to be individualized to the specific mutation that is either in you as a person with familial disease or in your lesion as a person with a sporadic disease. And that's way expensive and way weird just to have a clinical trial of one or of your family. And for right now, it means that every experiment starts, every development of treatment starts from scratch. We don't have a way to just cookie cutter it for people. And it still needs a viral vector. To deliver it, viral vectors can sometimes cause immune responses, which can be very dangerous and in some cases life-threatening. So what we're looking forward to is a platform so that we can cookie cutter it, so that we can create these gene-edited therapies very quickly for many, many people. And you can see here, this is one example. This little girl had, in just 6 months, a very specific targeted gene therapy made just for her. And that was due to a lot of pre-work done by a lot of folks in various research facilities around the world. And so this is what we want for everybody. We want, within 6 months, for them to be able to develop a treatment for you that is specific to you and that will cure you. And this little girl right now no longer has the ammonia buildup disease that she was born with that was going to take her life shortly. I think it could be a little boy. I'm not sure. In my mind, I have her as a little girl. We also want to replace viral vectors with alternative delivery mechanisms so that we don't have that immune response. And we want to bring down the cost. We've all heard about the $2 million gene therapy cost. We believe that once you platform it, it's going to be a much cheaper enterprise. There was just an announcement by an agency in the U.S. government, despite all the craziness that is happening right now in the U.S., and I won't even go there. There is a division called ARPA-H that has put out a call for just this. There's a competition right now that they're putting a lot of money into to encourage consortiums that include commercial entities and academic entities to create platform gene editing to replace viral vector delivery systems and to bring down the cost so that you can go to your local hospital and get your personalized gene therapy. So, last slide. The next, Helen, am I past time yet? Okay, the next 20 years promise a cure for cavernoma, tailored to your specific needs, genetic therapies for familial disease, targeted therapies for sporadic disease. In 2030, what we're looking forward to is focused ultrasound approved for adults. If I have my way, that's what's going to happen. We're also hopefully going to be looking at the first in human gene therapy trial. It'll be a CCM3 patient. We'll figure out in the meantime whether it's an adult or a kid. Clinical endpoints should be well defined by then. We've been working hard at that for almost 10 years. Targeted delivery experiments will still be ongoing. This is a hard problem. The field itself has not advanced far enough. And we should have AI-assisted early diagnosis in place. In 2035, so 10 years out, we will have focused ultrasound approved for kids. We will have an approved CCM3 gene therapy, fingers crossed. Precision genetic editing trials will be underway for the other familial disease, forms of the disease. We will have targeted delivery in 10 years for at least 3 drugs. Remember, I told you each combo needs its own trial. And we'll probably have some biosensor monitoring and AI in testing, not necessarily approved, but in testing. 2040 to 15 years out. Gene editing therapy for CCM1 and CCM2. Targeted therapy of at least 5 effective medicines, whole brain focused ultrasound for our familial folks, biosensor monitoring and AI will be driving treatment choices. Who gets what treatment will be determined by what is being detected by biosensors and what AI tells us makes the most sense. Microbiome modification may wind up becoming a way to actually control the disease. And then finally, 2045 and plus, patients will be diagnosed before they hemorrhage, and any patient can avail themselves of safe, permanent treatments, a cure. And that is what I got for you. Thank you. Thank you.